Antibody-drug conjugates are quite an interesting class of treatment, and we do actually have a license and reimbursement for Brentuximab vedotin, which is an anti-CD30 antibody-drug conjugate, so we're quite used to using that in the relapsed Hodgkin's and anaplastic large cell lymphoma space. There's a whole raft of new antibody-drug conjugates coming through though, which are quite exciting from a sort of molecule development point of view. An ADCT301 is an example, so that's an anti-CD25 antibody-drug conjugate. It uses special conjugation technology, so you know exactly how many drugs are attached to the antibody at any one time, and that's very different from something like Brentuximab, where really it could be anything from 0 to 8 molecules. We know that it's going to be 4 drugs per molecule, delivering a much more constant concentration to the cell. It's also got improved linker technology, and also the toxin, the warhead as people tend to call it nowadays, is an extremely potent toxin called a pyrrolobenzodiazepine, or PBD dimer. And that's orders of magnitude more potent than, for example, MMAE, which is on Brentuximab, or calicheamycin, which is on Mylotarg. So these drugs are very interesting sort of biologically. The trial that we've been involved with in the UK, it's a joint UK-US study actually, is this drug called ADCT301, and the eligibility criteria were simply relapsed refractory lymphomas. It was mainly T-cell lymphoma and particularly Hodgkin lymphoma patients actually ended up going on this trial, and we saw very, very high response rates, so over 80% in Hodgkin lymphoma, heavily pretreated patients, often relapsed or refractory following an autograft, Brentuximab, even checkpoint inhibitors. So very interesting efficacy data. There are certainly interesting toxicity signals as well. These drugs are not easy necessarily, so there's quite a lot of skin toxicity, some liver toxicity, and even some neurological toxicity, which we think is actually an on-target effect, possibly by depleting regulatory T cells. So, as with any phase 1 trial, it's been a learning experience of how to use these drugs properly, how to minimize toxicity, but we certainly look forward to developing that drug further, given its really very high activity levels.